The race to find the structure of the DNA, the molecule that has information in the form of subunits, a sequence of subunits, that is the nucleotides. Many labs across Europe and the US have started or kind of had an intense competition to find this structure. And it is a fantastic culture for science as well. There are interesting stories about how these competitions between the labs happen. Today we will discuss about Chargaff, Erwin Chargaff. I am Dr. BCMR, Assistant Professor at Shastra. Erwin Chargaff has published a very important paper that has kind of set the tone for the structure of DNA. Of course, there were many other scientists and studies before who have shown the composition, that there is a pentose, there is a sugar, and there are 4 nitrogenous bases and many other stuff like that. We are discussing about Erwin Chargaff's contribution here because it is at the juncture of what actually, how the inheritance of information happens, and that is why it is important. So he has published in Experientia. It's a very nice paper, which mostly it's about the chemistry, about how these biological samples are taken, degraded into their constituents, like just for the nitrogenous bases, and using chromatography and other chemistry based techniques has identified the proportions and also what they were. So first things, some of the things are interesting because, and also the scientific approach is nice. Say we would find about what are the bases that are there. We might end up finding these 4 bases. These are the 4 bases. Qualitatively, you can say it. For a reason that is unknown to me, Erwin Chargaff has gone to see what is the proportion of each of them. Is it same or is it different? That's the scientific thinking, probably. And he has designed these experiments in such a way, or he must have done many things and put them together coherently. The sample could be from one individual, and it could have been taken from different samples and then checked for the proportions of these 4 bases. Then he also had several other samples, say one human, it could be one from a bacterium, one can be from an ox, and then tested what is their composition, the nitrogenous composition of these nitrogenous bases. And these gave some leads, and that is what we are going to see. Some of the numbers, all the data is taken from the paper. So the sample here is from man, and he has tested from different samples, and that is sperm, thymus, and liver. In liver, he tested from normal as well as carcinoma. And as you see, first thing we need to try to compare what is the composition of adenine in all these 3 or 4 different samples. If we look at that, we can see that it is about 0.27, 0.28, which is, although the samples are different, some of them even that are cancerous, still the composition of adenine is relatively same. The same can be also seen for adenine, for cytosine, as well as for thymine. I guess some of the samples didn't work out as well, could not be determined probably. But we can largely agree that the composition of say adenine across all the tissue samples is almost same. And there is one more important thing that he has found, is that the composition of adenine is equal to that of the composition of thymine. The composition of adenine is equal to that of thymine. Similarly, the composition of cytosine or guanine is equal to that of the composition of cytosine. These are something very trivial for us, but at one point they were very significant information. So he also tried some bacterial samples, that is yeast, and also some mycobacterial samples. And what he found is the adenine and say all these bases are almost similar in these two yeast samples, and they are very different from that of the avian tubercle bacilli, which means that the composition of these bases is specific to tissues, specific to a species. One species has a specific nitrogenous base composition, and it will be different from that of other species. And there is something else he has done, and that is also an important factor or important point that we need to remember. He has also tried with ribonucleic acid, with ox, another example. This is again DNA, and he has found the same thing from different tissues, and he has found that the composition remains the same, and that A is equal to that of thymines and guanines is equal to that of cytosins approximately. Then he has performed some other experiments on ribonucleic acid, that is RNA, and that is something important for our understanding, is that say RNA, majority of the RNA is single stranded, and majority of the DNA in all of these species taken together, they are double stranded. The biggest variations, if there are any, it would be with viruses. In viruses, we may find some single stranded DNA, and we may find some double stranded RNA. So now that we know about the structure of DNA, it is quite easy to understand that anywhere there is base pairing, A with double bonds with double hydrogen bonds, 2 hydrogen bonds with thymine, and G with 3 hydrogen bonds with cytosine. These are base pairs. So anywhere there are base pairs, the rule of A equal to T and G equal to C will be maintained. With RNA, like we are discussing or shown in here in the table, we can see that the adenine is not equal to that of uracil, and similarly the G is not equal to that of cytosine. However, many times he has done that, you would always find them not so similar, and they were very different. So to conclude, in the first line of his conclusions in the paper is nice, and I would like that as a reminder for us. I quote, "Generalizations in science are both necessary and hazardous. They carry a semblance of finality which conceals their essentially provisional character. They drive forward as they retard. They add, but they also take away." So from his paper, he was being very balanced about what to take from it and what not to take from it, or he is being philosophical in a way. So to conclude in his own verbatim, he has come about with DNA from animals, and microbial cells contain varying proportion of the same nitrogenous bases, that is ATGC, and their composition appears to be characteristic of species. That means one species will have a certain ratio of these ATGCs, but not of the tissues, which means that, to say that all the tissues of one organism will have the same base composition. So the base composition wise, we will remember them as purines is equal to pyrimidines, the adenine is equal to that of thymine, and guanine is equal to that of cytosine. Except where they are single stranded, either it is a majority of the RNA and some of the DNA. These do not follow these Chargaff rules, but every others will follow. One thing we have to remember about RNA is that RNA is single stranded, but it can have intramolecular these base pairings. So in this region it may form a base pair, and in that region the composition may match it, but the rest of the regions do not base pair, so they do not follow the Chargaff's rules. Thank you very much. We'll see you in the next lecture.